All right, so you see this slide already. Now, how do you extract those features? There are a number of ways in which the brain puts the activity of multiple sensory neurons together to extract features. I want to give you first a sense of what is low-level processing. And in low-level processing, you basically combine low-level activity of sensory neurons in what we often call the ganglia. We're going to talk more about ganglia in the future. The places where the first magic happens, where you're putting, for example, together the activity of many Pacini corpuscles, or when you're putting, or when you're combining the activity of different types of neurons. For example, one pre-processing feature that is very basic in a visual stream is, and what we find in the lateral geniculate nucleus and in the which is in the thalamus, or in the optic nerve itself, is the notion that cells in the retina, or areas in the outside world, can be encoded via presence or absence of light. Now, one thing is clear, right? So if you have, like, if you live in a dark world and you have a little bit of a white dot in one point of the world, your retina in that area is going to be activated. And that is basically already encoding the feature of location as a function of the entire visual field. So basically already there's information, spatial information there. But beyond that, if you do the opposite, and if you have like an entirely white field and like a black dot, there is going to be also a neuron in that position saying, well, that place is black. So you have that ability to distinguish light from dark in that way. And this is done via the so-called on and off cells. In on and off cells, and this is what I am missing here, I just noticed that one of my YouTube links has been taken down. So the link that explains this particular arrangement in the retina from cones to horizontal and bipolar cells has been removed. I have another version of it that I'm going to share with you later. But the nice thing about this one, it was just like a 2-minute like short snippet. Unfortunately, is no longer required. I'm not going to do the search right now, but I am going to do the search later so that you can find it. In any case, the idea here is that there is a little bit of a circuit at the level of the retina that already is able to extract whether a point in the visual field is lit or not. So we call, for example, the on-center cell is a cell in the lateral geniculate nucleus or in the optic nerve that will respond to light in the middle, but only if there's brightness or there's darkness around it. If it's completely light all over, it's quiet. So it not only responds to the pixel, but it responds to an isolated pixel in that particular place. You have the convert of the cell, the opposite of the cell in the off cell. And the way in which it responds, we can play with here. Now I hope that this link is still working, and I'm going to pump the link into into the chat in Zoom so that we can all play together. There's a little bit of a background, and there's a couple of instructions on how to use it, and there is an illustration of how it works. So this is the stimulus you can create, is the dot. You can put the dot in the center, or you can create a light stimulus that has an edge onto the receptive field of a single neuron. So here we're looking at the activity of the receptive field of a single on and off cell. And so depending on whether the cell is an on cell or an off cell, it will respond to a dot in the center. So what I want you now to do is to play with this visualization and then tell me your observations. You have 3 minutes, if that's all right. I am expecting you to be able to open this. If you aren't, then we can also break out rooms. You finished? 3 minutes. Actually 2.5. But I think that this is relatively straightforward. I just want us to go through a couple of these quiz questions. So can you read the quiz questions in the slide that I'm sharing?
Yes.
I will poll you via the chat, okay? So you answer via the chat what do you think that the answer is. When I have about 5 questions, I'm going to click 5 answers. I'm going to use that to click the majority. Shall we? A neuron with an on-center receptive field would increase its firing rate when? Too easy. I have to submit before. This is a very old little presentation. Center surround receptive fields are the earliest stage of... Oh, you haven't read the introduction potentially. Doesn't matter. I want you to think about this now. All right? Click submit. Let's see if we're right. We're right. A neuron with an off-center receptive field would respond maximally to A. Have you already answered the new one? The majority, oh, now we have a little bit of disagreement. D seems to be winning. There was a C and a single E. So I'm going to submit that one. Ta-da! Did this surprise you? I mean, it's so easy. You have a question? Any of you have a question, or you want to think about the answer? Yeah, you do. We did have someone answering A. Yeah. Okay, guys, you will have this that you can go through the entire quiz, and you can also read the instructions. But now what I want from you is more fun. I want you to tell me, how would you make, say I give you 2 types of neurons. I give you the input neuron, give an input neuron, and I give you the ability to, actually, let me phrase the question differently. How does this work? I mean, like, how does the on-cell work? I mean, like, can you share with me like an intuition about how does this work? What is necessary to make this work?
So the question is how this on-off cell works specifically.
How do you, how, because an on-off cell is not the retina cell. The retina cell is the cone. The cone is going to respond when there are photons coming in, and it will respond with a hyperpolarization that is going to be transmitted. But that's not the question. The question is, from a theoretical perspective, how can you actually achieve this type of behavior? Like, just thinking about how would you make it?
Expose the cell, or if it's a single cell recording to a stimulus, and then, or just I don't theoretically speaking, expose the perceiver to the stimulus. And then
Maybe not in an experiment. So, like, get the word theoretically out. Say you want to make something that works like that.
To tell it then if it's
You want a neuron. You want a neuron
Yeah.
that responds positively when the inputs to the retina have the center illuminated and the surround dark. Let's start only with the on cell. The surround needs to be dark and the center needs to be bright. How would you find out whether there's a difference, whether the center is bright and the outside dark? You need to create a comparison.
Yeah,
right? And you need to create a comparison between what is in the rim and what is in the center. And how do you create a comparison in neurons? Well, maybe you have to inhibit. Is a word that we're bringing in anew here. I was just wanted to see if this word was latent in the distribution of you, whether you would come up with the idea that, well, you need at least two cells, okay? So first off, you need at least two more cells. You need one photoreceptor that is looking at the center and one photoreceptor that is looking at the rim. And then you need to compare their activity. If the activity of the center is larger than the activity of the rim, then we allow that information to pass. So we need some sort of subtraction between two inputs. That subtraction is conducted by the horizontal cell. And what you have actually is pretty much this. You have surround cones and center cones that are being mapped onto a horizontal cell and a bipolar cell that subtracts the information. So this video I'm going to provide to you so that you can review this information and you can think about this a bit more. But I want you to think about this first in detachment from actual complexities of spike production, more from the perspective of the mathematical necessities. If you want to find contrast between two things, the one thing you have to do is to subtract. Subtracting is something that you obtain with a neuron when you have an inhibitory input. So ultimately here you will find something that is an inhibition from the surround to the center, and vice versa, the opposite too, to obtain the off-on cell. Now there's another thing that you're thinking about. Okay, so the center cell is less input than the outside cells. So if you want to compare them, you will have to balance them out somehow, right? By, for example, emphasizing the connection of the center cell to the horizontal cell, which is eventually going to become the optic nerve. And then in the optic nerve, you will conduct already the information that has extracted the feature of edginess or of contrast. I'll send you the video, we're going to look more about that. Now, let's think about another property. Let's think about motion. We open this link here. Say you have a bar from the left field to right field, and you want one neuron in your brain that is going to extract the feature of a direction of motion. So it will only fire if something goes through it at a certain velocity. So, what are the things you need? In order to extract motion, can you do motion with a single cell, for example? Can you do motion if you have a single input cell? No, right? You need at least some sort of way to represent the fact that something is going from one place to another. Preferably, you would like those guys to be close to each other. And you will have to somehow make another neuron active only when a certain motion obtains. So if you have, for instance, the motion of this white bar here in your visual field, you have two neurons. The first neuron is going to see that object first, and the other neuron is going to see that object next. You need a downstream neuron that is integrating the information from the two upstream neurons, right? And you need some way to encode the fact that there is a time between passing on to receptor 1 to passing on to receptor 2. Motion detection is believed to be done via a structure that we call the Reichert detector. And the Reichert detector is a very simple circuit with two photoreceptors that looks a bit like this. So you have neuron 1, you have photoreceptor 1 and photoreceptor 2 in your retina. You have a connection that goes through a little block here, and I'll tell you in a moment what the block is. It's a tau. Goes into another neuron, and this is represented as a circle. So the information from that stimulus coming over here is going to percolate through this connection. This neuron down here later on will receive also the stimulus from this later accessed photoreceptor, which will converge in this guy. If the two neurons produce the spikes immediately, then the downstream neuron is going to hear one spike, then another spike. But if you want to localize the motion, what you want is to know when the two of them are active with a certain delay between them. This is the delay, the tau. So you have the information converging from two photoreceptors onto a coincidence detector that is doing the pre-processing extraction. It's extracting the feature of motion. It's pixel-based. You can do this in the entire visual field. This is an array of photoreceptors here, and every single pair of photoreceptors can judge local motion as a function of their neighbors in these integration neurons. So that guy is receiving from this guy and this guy with a certain delay. And it will be summed over a big, maybe like one big neuron then takes information from all of these small neurons that are saying, say, motion from the left to the right, becomes selective, that downstream neuron becomes selective from motion on the visual field that includes many pixels at the same time. A Reichert detector. That's a model of motion recognition that we know now is very close to what happens in the fly retina, and that we know now is very close to what happens in our retina when we think about the delay mechanism as being a calcium signal. So this is very cool. So we have an abstract way to extract the motion feature, and I wanted to then quickly go through this so you can then, with a little bit of patience, open this link, and I will do so, where we see a Reichert detector, actually 3 Reichert detectors, each of which selective for a different velocity. On the right graph, you see the activity of the center neuron for the intermediate high shadow detector, the one that's here in the middle. And you can, if you make the stimulus go fast now, or super slow, it will activate the neurons in differential ways. In this case, it won't activate, like if the stimulus is going too fast, it's not going to activate the initial neuron because the neurons have different delays. I don't know if you can see the green line growing, I hope you can, but the green line, can you see a green line growing? That is the delay, effectively the delay. And there are many, many ways you can implement that via neurons, of course. One thing you can do is you can make an axon that is longer, or make an axon that is less insulated, for example. Or you can, in fact, make that particular neuron respond a little bit slower to the stimulus because it's an integrator. It might take more time. So the general notion that I want to convey here is simply how a certain structure implies a certain feature extraction. Is this clear to you guys? Okay, circuit-wise, this looks almost like an SR latch. What's an SR latch, Mace?
It's a component in like computers we use.
Cool, right? So necessities in the universe have been used by nature too. And so we do things as components like this because ultimately, if there are certain features where we want to extract, I want to impress upon you the idea that there are certain necessities. Now, how exactly the nature implements it might vary from time to time, but the kind of way in which we use these things can analogically be used in different systems. And your example, Maze, is perfect for this. I wanted to tell you that it's not only in vision that you need direction recognition. Also in touch, you have direction recognition. And that also works in the same way, by necessity. It has to go from here to here, and there has to be a certain encoding for the speed. And also, there are limitations on the system that relate to the actual structure. We can't see all possible velocities in our visual field. If it goes too fast, we don't see. If it goes too slow, it doesn't appear as moving. So there is a spectrum of pre-processing that we have been ecologically attuned to absorb. So the monkey hand also can discover what is the direction of motion of a tactile stimulus, and the way that is thought to happen is related to the high shadow detector. Simple enough, right? So now we talked about two very simple structures that can extract specific low-level features from stimuli. Let's talk about how they're used a bit. Here, this is an example. This is an experiment where we have dots in the visual field randomly moving in the visual field of a monkey, and we want the monkey to make a decision about whether the motion is coherent, moving in a certain direction or not. Right? Have you been exposed to this paradigm before? So we then select a subset of the neurons to make moving in the same direction. In the left, or in first part of the experiment, all of the random dots are moving with random directions. In the second one, 50% of the dots are moving with the same direction. So you can see that, like, amidst all the noise, there's a field of dots going upwards. I could actually have a video here. Or all of the points are going up. I mean, this gives you a very powerful visual illusion that there is some sort of like two layers. One layer that is separate from the layer of the uncorrelated neurons that is moving upwards, for example. Now we asked the monkey to do this. The way in which he has to pre-process those features is by pooling together a number of neurons that are recognizing specific directions across the visual field. The reason why I bring this to you is for you to think about the population encoding that is required here. We need many neurons deciding what the motion is in specific parts of your retina. So if, like, I'm thinking about your retina as everything that you're seeing right now, we need decisions about the motion in many different places. That's the idea of parallel distributed processing and population encoding that we're going to be drilling a lot in the near future. But for the moment, what I wanted to get across is the notion of feature encoding. The monkey can do this task, by the way, and it has a funny thing here. The monkey makes more incorrect decisions than we can extract of a single neuron in his IT. So sometimes, sometimes you can find a neuron that knows more than the monkey. I don't want to go there yet. We're going to talk about decision making a little bit farther down in the future. But for the moment, the only thing that I wanted to convey is that this decision about whether the motion is coherent or not has to pool together the information about a number of lower level feature processing neurons. Now, the last example of feature processing for today, I think you will find quite obvious. So, say this is your retina. This is a pin art, okay? So you're familiar with pin arts? So say this is your retina looking at my nose. Okay? You have a bunch of photoreceptors, and obviously we talked about, that there's some local processing, but let's think about them being activated by my nose. And the amount of activation, for example, is the depth of the pin or whatever. Now, you want to find an edge on this picture. How do you go about it? It is not by mistake that I have the slide on the bottom. I really want you to think about local pooling of retinal neurons that are extracting certain geometrical features of the environment. This brings us back to why Hubble and Wiesel see the things that they see. All right, there's also some noise in the encoding. If you take a local neighborhood of retinal cells and you arrange the connectivity of a downstream V1 cell, in this case, as a set of on and off cells, in this case on cells, you can encode a luminous bar on a dark background. With me here? Now this done. Edge detection for you. How do these guys come up to do this? I mean, like, do you need, for example, to learn, like, as say you're just born, are these connections learned? Yes, they're learned. Orientation detection is a process of self-emergence given natural images via Hebbian learning, amongst other things. But now I finally get to the point where we can talk about our first feed-forward neural network. I want you to create do-it-yourself receptor fields. Project number 5 builds on project number 4 because of connectivity, but is also rather independent of it. So if you didn't do project 4, is all right. It just gives some intuitions about connectivity. But what I want you now to think about is the following. And let me see if my QuickTime is now working as I would like. Yes. So you can see this, right? No. I have two colors. So say this is your retina, full of photoreceptors. Say you have an orientational stimulus. that is occupying a part of your retina. Project 5 is about building a simple feed-forward neural network that will detect oriented bars, irrespectively of how they are oriented. Just so happened that I should have all in different layers to be able to rotate. But you can get the idea here that this bar can be oriented in different directions. You with me? Now this is simple enough, of course. Now we have, in the project, you will do this for 3 neurons, and you will assume that we are dealing exclusively with on cells, and you will assume that your on cells can have the activities of either 0 or 1. 0 if it's black and 1 if it is bright. They represent the clamped activity of your cones, or indeed lateral geniculate nucleus. We're working here on the level of representation. So your input neurons can have values of 1 or 0. Each of the neurons can have a value of 1 or 0. Then you make a network, and you're thinking, "Oh my word! I mean, like I'm scared. I mean, like what are you going to make us do? We just learned about, barely how to make some spikes, and how are we going to do this?" Don't worry. Just be with me for the moment. So you have here a neuron that is receiving from all of these inputs. And I want that neuron to tell me whether it's receiving a stimulus that represents an oriented bar in that input. That neuron is going to respond with a 0 if it thinks that the bar is not oriented in that direction, or with a 1 if it is. Now, how you're doing the mapping between the weights of the input neurons onto the output is important. And that's why the feature processing in the brain is so related to topography. And that's why I was drilling the idea that it really matters here that this neuron is a neighbor, so that this neuron is a neighbor to this neuron. Right? It really matters. And it really matters the order of the neurons that, for example, are reading a particular orientation of the bar. So, how are we going to do this? We're going to do this by using a simplified model of a neuron, which is the McCulloch-Pitts neuron. And McCulloch had a great idea. He thought that what neurons do is to recognize patterns. And in 1953, he was working together with biologists to try to derive a couple of principles on which they could try to see whether they could make pattern recognition be the case with simple neurons. In fact, they wrote a paper that is called The Logical Calculus Immanent in the Laws of the Brain. Let me see if I... so I believe that I have. So this is the original paper. A logical calculus of the ideas immanent in nervous activities. It is 1943 we're talking about here. And we still use to this day McCulloch-Pitts neurons to do pattern recognition. We now do a little bit more than that, and there are certain very strict assumptions that it makes about how things work. And I'm telling you about the assumptions as we go. But we will use his ideas from 1943 to create a receptive field, and also to expose more problems and more ideas in this entire field. Now, I want the way in which the inputs from the input neurons are mapped onto the output neuron. We say that they're making synapses, and that these synapses have different weights, okay? So we have 3 by 3 inputs, and so we will say that we have W1, W2, W3, until W9 input neurons. They are going to be the synaptic weights of these connections. And these are numbers. And these can be numbers, say, to make it easy to start with, let's say that your number is going to go from -1 to 1. You can select any numbers from -1 to 1 for the weight matrix of this neuron. Now, you're thinking, how do you map the topology? So this, if this is now neuron 1, this is neuron 2, but you could also make this guy here neuron 1. Well, do it in a way that is intuitive so that you actually know which weight is related to what. But know also that what is important here is the topology of the connections that is transmitting information about the topography of the input. Let me make the distinction between topography and topology quickly. Topography is the idea that you have a one-to-one metric map. Topology is the idea that you have relations and relationships in the connections. So you have a certain topology of the network and you have a topography of the stimulus, all right? These terms will appear again, but topography is at the level of the stimulus, topology is at the level of the network. So you have 9 weights and you have an input stimulus with the neurons of the input actual. And these are the activations of the input neurons. Okay, so you have 2 vectors. You have the input vector and you have the weight vector. Simple enough. You have 9 neurons that multiply by weights and sum into the downstream neuron. So you're going to do what? What is called the weighted sum of the inputs onto the neuron, and you're going to select what the weights are so that that particular oriented bar is recognized or not. There's one more thing. What is the thing that is missing? Can anyone tell me what am I missing so far? Because I said that I want the output of the neuron to be between 0 and 1.
Can I ask one question?
Sure. You can ask many questions.
Okay. Everything is clear to me until you went writing like the x1, x2 in green. That's what I don't understand.
Okay, so you understand here that I have inputs, right? So these x1s represent particular. Let me actually put this layer. Let me use another layer. Hopefully this is going to be on top of everybody else. No. So this neuron here, let's say it's active. It's neuron 2 of the input. It's the second photoreceptor. It will say that this in the weight matrix, or sorry, in the activity vector of the input, we'll give it a 1, all right?
Yeah.
Is this clear?
Yeah, it is.
And then, if we have these 3 being activated because they're receiving a black bar, so this guy is going to be a 0, 1, 2, this guy's going to be a 0, this guy's going to be 0, this guy's going to be a 1, 0, 0, 1, 0.
Oh, so a 0 if it's not reacting, and a 1 if it's...
Exactly. So the 1 was when the photoreceptor is bright, and the 0 is when it's dark.
Oh, okay.
Okay, so I said that you're activating the photoreceptors in a way that we have a 1 for bright and 0 for dark. So it's becoming very trivial to solve this problem, but there will still be insights from it after you do it. There's a couple of linear algebra aspects that I still haven't talked about, and the one thing that is missing, you still haven't told me.
So a function to go back from the sum to a number between 0 and 1.
Exactly. So a step function, for example. Let's see if everybody can understand what this graph is saying. maybe I should do the function in a certain color. Function of input, no? So a function of x. Can you guys read this function? What does this function say?
Doesn't this mean that as soon as there is a signal applied to the neuron, it immediately fires? Because the threshold seems to be put at 0.
Remember that the weights can be selected also.
-1. All right. So as soon as the threshold of 0 or more than 0 is reached, it will fire.
Exactly. Now, will fire means it will be 1. Whereas we've been talking all the time about how is it that the neurons have all this exquisite set of firing patterns. Keep that contradiction in your heart for the moment, and let's resolve it as we go forward. But for the moment, let's talk about being active and being inactive. McCulloch-Pitts was actually talking about a spike, but obviously we're talking about something that happens through time. So we imagine this as being clamping an input. So we present the input, and the answer of the neuron will be I'm active or I'm not active. So this is the Heaviside function. Now there's another, and we often represent it with an h of x. So this sum goes through an h of x to the output that is basically a 1 or a 0. Now, a Heaviside function always has a threshold in 0. There is a term that you might have heard from previous studies that you will also see in the project, which is called the bias, but I think it adds an unnecessary level of complexity at this point, and so we're not going to talk about it for the moment. It will be already in the project. I'm going to make the project available immediately. It is do-it-yourself receptive fields. And it will take McCulloch-Pitts units, and it will walk through a little bit of the history and some conceptions of the McCulloch-Pitts units, so that you can make neurons that select features, select orientations as features, basically respond to oriented bars. We're talking about a area of visual cortex that is a small patch of local field. So it has a certain aperture. The aperture is the angle that the visual field has. So if this is the image of the world in your retina, we usually talk about the size of things as a function of the size in angle of what it contains from your retina. So basically the aperture is the angle of an image contained in the retinal projection. Maybe I should start again. So if this is your eye, and this is the house, and the house is projected on the retina, the aperture is basically a small patch of retina that maps onto a little angle in the world, alpha. Just to bring you to the notion of aperture, if you have never taken an analog photo before, but it's not essential. The point here is that we're looking into a small patch of the retina, and blue here, the light blue represents the retina. Okay. I'm going to make the project available right now, and we're going to discuss some other implications of this, and we're going to start assembling our understanding on how we believe that feature extraction builds onto meaning. And many of you already feel it in your hearts where this is going. We're going to flesh out more of this, but I couldn't put more emphasis on the fact that it's important to understand how weight matrices look like with respect to the stimulus that they're selecting. And so the main goal of this exercise, giving you intuition of the relationships between network topology and feature selectivity. So the idea here is really to give you an embodied sense of how a set of pixels represents a nose, and so forth and so on. Let me see. I'm going to make the project available now in Canvas, if it hadn't been.
Do we need to finish it by Tuesday?
I would be happy if you did.
Together with project 3, I believe.
I'm going to delay the project 3. Well, there's no delivery. Yes, ultimately yes. Actually, the DIY receptor field already is here, so you can already go to the project. Why is it not? Let's see. Let's see. Open with Google Collaboratory. Just see if it's the right one. Yeah, it is the right one. Okay, guys. So let me share some screen here. The project 3, so the spiking networks project is very dangling. It doesn't really matter when you finish it, because ultimately the insights from spiking will be coming from from a set of different perspectives. And the essence of project 3, which is the idea that there is equilibrium between inhibition and excitation for certain spontaneous firing, is something that I want you to acquire, but it doesn't have a given deadline. I've given a deadline just because orientationally, but take the projects and do them as you manage. When I actually put an assignment date is the orientational idea on how long you should take. None of the projects should take more than 4 hours. None. Even for a non-expert coder, especially because they already have the solutions. So probably what I request is to read the project until next Tuesday and see if you understand solutions. But I really think that this is within your spectrum to do. Recommendation: working groups from now on. Go into Microsoft Teams, find a couple of cool groups. And group yourselves. Just basically drop in one of the team regions, and I could break out rooms here too, and then those would be rooms. I will break out rooms here. What would you prefer? Random assignment? Because you guys don't super well know each other yet, and I just want to know, would you rather have some random assignment for breakout rooms, or would you rather assign yourselves? Any opinions on this?
Outside of the meetings, I work together with some people already.
Yeah, so I think that it's all right that you just because it doesn't, the size of the working group doesn't matter. It matters that there are working groups. So if you go into a group that is already working, I can't see the study groups anymore. Is there no Dropbox somewhere there where the channels are? Can you click on the channels maybe? I mean, I'm sure that they have.
Then I only get the organization one.
Ooh. That is strange. I'm going to work group 1. Apologies, I don't know what happened. Any of you guys deleted the teams by mistake? I don't know if you had the permissions to do that. I'm going to make 3 work groups, and if more are needed, just let me know, okay? So I just added the channel. Can you see? It's called work group 1, work group 2. Okay, now we have 3 work groups. Assign yourselves, meet new people, listen to what they have to say, see if they're confused. My suggestion now, before you go in, I'm nevertheless going to give you the opportunity to find yourselves in a breakout room in Zoom. I'm going to make some breakout rooms now. And you can discuss the project and call me in if you don't know what you have to do, or if you're completely blocked. But I think that the project should, I hope, be very self-explanatory. We will be assembling on those notions as we talk about decoding in the next lecture. But this is a very very crucial point in which you first encounter your first feed-forward artificial neural network. There's a lot of theoretical aspects about this that we're not discussing yet. For the moment, I just want us to have a notion of what weight matrices should look like. Okay, I will now break up the rooms